And now that you have learned two important secrets, you have learned the secret of the gut. Okay, and now there was a wonderful maestro on the stage who also taught us about the hormones. So now it's time to get into the blueprint. So, what's the blueprint of the body? What's the blueprint of the body? DNA, genes? Yeah, genes. Do you know how to unlock the genes? Well, we're talking about the genes. Anyway, now we as functional medicine practitioners, we definitely know that. Can I help you? I'm sorry, I won't be able to give a second. All right. Yes, we're talking about the genes, and genes need to be expressed. If they are not expressed, you know, genes are not our destiny. This is what we keep understanding from functional medicine, isn't it? Yeah? That's not our destiny. Is it? Anybody believes it's our destiny? Definitely not. We as functional medicine practitioners, definitely not. And the person who's going to talk about this, he himself has led this life initially that thinking that genes is his destiny. But he changed the entire world, not only his world, but the world of the people whom he teaches and whom he touches. Methylation, his favorite topic. And I'm pretty sure the fans of Dr. Musa definitely know about this as well. That he loves this topic because this was the topic that changed his life. And that's why this topic cannot be better expressed by another, that the one person from the functional medicine family, very much loved by everyone. Put your hands together for Dr. Anish Musa.
Thank you very much, Amarjit. And thank you very much, audience. This is our fourth attempt to do a functional medicine conference, but this time we are doing an on-site. So thank you very much for joining us here. By the way, Dr. Preeti is doing BHRT courses. If anybody interested in joining the course, then you can learn more about the BHRT. So please contact Dr. Preeti about that. Okay, guys, let's start our today's topic. I will talk about the methylation. Okay, do you want to add 10 years in your life? And that 10 years could be active, more healthier than what you are living right now. Yes, then we have to unlock the secret of methylation. We have to learn the methylation, what the methylation is, and how methylation can help. At the end, I will also talk about how to optimize the methylation. Because methylation is the process, central process in each and every cell that can be in your brain, that can be in your hormone metabolism, that can be in your detoxification. And of course, that can help in cardiovascular disease, right? So let's learn the methylation. So all we talk about the epigenetics, right? So we talk about the environment is affecting our genes. So in short, we are dealing with the environment, right? So this environment is going in our cellular environment and then it goes in our DNA, right? And then DNA start expression of that particular function. So you have an environmental trigger, then you have genetic expression, and then your genetic expression leads to modification of your cellular or biological function. Let's take a simple example. We all know insulin resistance, right? There are many genes they play a role in insulin resistance. And we all know that environment and lifestyle is affecting our genetics. We know how. What is the primary factor that leads to the expression of genetics? That's the methylation. And methylation is basically our chemical CH3 group. Now, if you have a healthy expression of genetics, you have health and wellness. And then you can achieve the longevity, what Dr. Ashwini told. So that's really expression of genetics. If the genetic expression is bad, then you will have a dysfunction and finally you get the disease. So that the central process is methylation and methylation can affect all your body function. So if your patients or yourself is suffering with any kind of cardiovascular disease like myocardial infarction or atherosclerosis or your patient is suffering with any kind of hormone dysfunction, what Dr. Priti told, maybe it's a hypothyroidism, maybe it's a estrogen dominance, or your patient is suffering with any kind of autoimmune condition or immune dysregulation or a chronic inflammation, or your patient have any kind of neurological condition like Parkinson's, Alzheimer, depression or chronic pain, fibromyalgia. So if your patient is suffering in any of these things, then central connecting dot is methylation. Because methylation is central process. And that central process is just a shuffling of our CS3 or methyl group. And that methyl group is one carbon with three hydrogen. That's a central chemical. And with all cellular process, we are actually shifting the CS3 group from one molecule to the other molecule, from that molecule to the other molecule. So that's the central process that happening in your brain, in your liver, in your kidney, in your cardiovascular disease, in your immune system. Each and every cell need methylation, and your DNA need methylation. So methylation help in phase 2 detox pathway. That's called methylation detox pathway. Then it's also required for the hormone production and detoxification, neurotransmitter synthesis like dopamine, serotonin. It also help in DNA methylation. That's called a gene expression, what we talk about. So one thing I would like to tell you is that if you have a optimal methylation, you will not have a gene expression. Like if you have optimal methylation, you will not have a bad genetics expression. Let's take an example. Let's say your patient have a family history of diabetes, means we know the family history that patient have a, some genetics for insulin resistance, right? But if we have a stabilized genetics, like with a proper methylation, so they will not have a genetic expression of insulin resistance in spite of they have an environmental trigger, right? So if they have an environmental trigger, and one patient, they have poor methylation, that genetic stability is not there, that bad genetics will express and they will develop the disease. Worse is, if somebody having an optimal methylation, that genes is stabilized, so they will not have an expression of that genetics in spite of having an environmental trigger. And that's called proper DNA methylation. So most of patients, they walk in my clinic, they have a under methylation. And that's why they have a chronic condition. So that's why we have to understand the central process in cell that can lead to a proper DNA methylation, right? And then of course it's going to play in energy, so the mitochondria, and then finally it's going to stabilize your cellular signaling, and that's called methylation. Now look at this complex slide, right? So you can see each and every body functions. There is a one methyl group from here. It goes to the methionine. Methionine going to convert to SAMI. SAMI going to convert in again homocysteine. Homocysteine goes again in cysteine. Right? So we are shuffling CSC group from morning to evening. Each and every second, your cell is shuffling methyl donor or CS3 group to all the process that's required for neurotransmitter, that required for hormone production, that required for immune function. But I will simplify it for you. Just focus on a one important cycle that is called folate methylation cycle. Now, if you look in central part, and I want to emphasize on homocysteine, look at the central part, that's the homocysteine molecule, and it's also anti-aging biomarker, I can say. So what homocysteine is? Homocysteine is amino acid, and then homocysteine get converted into methionine that you can see on the top. And other name of methionine is methyl homocysteine. So homocysteine get converted to methyl homocysteine, and that's an important chemical or biochemical step that requires one enzyme that is called MTHFR. So MTHFR stands for methyl tetrahydrofolate reductase. That's the enzyme that's do a conversion of homocysteine to a methionine, and that cycle goes on. And the cofactor we require is folate. Do you know why we have a vitamins and mineral? Why we are talking about what the main functions of vitamins and minerals? Why we need them? So vitamins and minerals, they are basically a cofactor of any enzymatic activity. So I told you about the trigger, environmental trigger that goes in the genetics. We have expression of genetics, and then what we do? We produce molecule. And one of the molecule is your enzymes for converting one molecule to other molecule. We require enzyme, and this enzyme activity required cofactor, and all the vitamins are cofactor. Like majority B complex, you can see they are cofactor. Zinc, magnesium, and other trace mineral, they are a cofactor. So in this process, what we had learned, the homocysteine get converted to the methionine that require a cofactor as a folate. And that converts homocysteine to methionine. Now just imagine that if this conversion is not happening because you don't have a methylation, poor methylation, so what will happen? You will increase homocysteine in your cell. Once you increase the homocysteine in your cell, that homocysteine will release in your bloodstream, and then you check in the lab and you have high homocysteine. Homocysteine also get converted to a cysteine, and the cofactor is vitamin B6. But right now, focus on a homocysteine. That if you have poor methylation, you will not convert your homocysteine to either cysteine or methionine. That's why you have an increase of homocysteine in your cellular cell, and then you can detect homocysteine in your bloodstream. So increased homocysteine means poor methylation. That's the primary clinical understanding of all the processes what we had seen. So understanding of this slide is just an high homocysteine means poor methylation. Poor methylation means poor genetic expression. And you have expression of wrong kind of genes, and then you have a dysfunction and disease you will develop over there. So this is simple cycle of folate and folic acid versus folate. So all in conventional science, and when I learned my functional medicine, I only know the folic acid, and all the doctors and gynecologists, they prescribe the folic acid to all the patients. But is folic acid is vitamin vitamin B9? Folic acid is actually a synthetic version of vitamin B9. So folic acid has to be converted into active folate, and that required enzymatic step, and that required methylation. Just take a simple example. Let's say your patient have a B12 deficiency and folate deficiency, and you prescribe folic acid. What will happen? So when you prescribe folic acid and the patient have a B12 deficiency, folic acid has to convert it into active folate. That required methyl donor. That requires CS3. So how body can get the CS3? He has to take the CS3 from B12. So when you prescribe folic acid, your patient have further B12 deficiency. That's why these minerals and vitamin, they work in synergy. That's why I always prescribe the B12 with the folate, because they work together for as a methyl donor, right? So if you look at this slide, the last step that folic acid need is conversion to the active form that required MTHFR gene. Now, if your patient have a MTHFR gene variation, what will happen? This step will be slower. So if this step is slower and your folic acid is not converting to active folate, what will happen in your cell? You will have a raised folic acid that is inactive, and you have a reduced form or reduced active folate that is active B9. So in short, you have an inactive B9 and you don't have an active B9. That means you are recruiting employee, they are not working. They are just occupying the seats, but they will not allow even the active employee to work. That's why folic acid is also harmful. If you prescribe the folic acid to MTHFR patient, they cannot convert this folic acid to the active folate, and in that case, the folic acid is harmful. So even we cannot prescribe folic acid to each and everyone, right? So that's the basic understanding of methylation cycle. what I told you, the anti-aging biomarker, as per my understanding, is homocysteine. So if you're not checking homocysteine to each and every patient, then you are doing a primary mistake, because that's the primary process that is happening in each and every cell that connect the dots to all your chronic conditions. That's your homocysteine marker. Another mistake we are doing in conventional science is we are looking at the very wide range of all the biomarker, but I will talk about the homocysteine. Now, if you look at the conventional medicine range or conventional lab, then the homocysteine marker is around 14 or 15, they say normal, right? So in functional medicine, we not talk about the normal, we talk about the optimal, and we talk about the wellness, and we want to talk about the anti-aging. So this range will not work when we look from that perspective. When we look from the perspective of functions, and we look the perspective from the anti-aging, this range will not work. So what is optimal range of homocysteine? Optimal range of homocysteine is less than 7. Ideally, you have to maintain your level less than 7. That is the optimal range. And once the level is optimal, that means your cell, all the cells, they are working. Let's take an example in this case. In this case, 14. So as per conventional range, this is normal. But in functional medicine, we not consider that normal. We consider this as a optimally high. And then we look at the methyl donor. By the way, B12 and folate, they are actually methyl donor. So if you look at the correct name of B12, it's a methylcobalamin, methyl. So you look at the folate, the methyl folate is the correct name. So they are the methyl donor. Now we know a little bit biochemistry from the earlier slide that we require methyl donor. And we know that if the poor methylation is there, patient have a high homocysteine. Right? In this example, you can see the homocysteine level is optimally high and B12 folate is optimally low. So we can say the homocysteine level is actually because of we have a poor methyl donor in our food. So it's a nutritional deficiency that leads to high homocysteine. So we can say it's a functional low homocysteine level. So that is very important. In the next slide, I will show you that my patient have a high homocysteine that is 29. You have seen many patients that homocysteine level maybe 20, 30. I had seen in my practice the patient homocysteine level goes to more than 65 in certain cases. But here you can see the patient's level is 29. Same time you can see their folate and B12 level is nearly optimal. So now we can say they have a proper methyl donor from the food, but still they have a high homocysteine. What could be the reason? So I told you the steps. We required enzymatic activity that is called MTHFR. So if you see this kind of result, if you can see this kind of lab, your suspicion should be patient have an MTHFR gene defect or gene variation. Even you can predict whether they have a homozygous or heterozygous of MTHFR. So what is homozygous and what is heterozygous? Homozygous means that both the genes are affected, and heterozygous means there only one copy that is affected. MTHFR is commonly understood and commonly tested gene variation that you can find out in methylation. But of course, that is not the only one genetics or only one genes that playing a cycle in methylation. There are other genes playing a role, MMR, MTR, COM. There are other genes also playing a role in methylation cycle. But the most important that we see variation in Indian population and around the globe is MTHFR. Almost 30 to 40% population, they have a genetic defect. Either it could be a heterozygous or heterozygous. But again, what I told you that the gene is not creating your destiny. It's a gene expression, right? So you have an environmental trigger, you have genetic expression, and then you have a disease, right? You can change the expression of your genetics. And I told you with methylation, you can do genetic expression and change of genetic expression. So in short, we have to look at the MTHFR defect if you find out the heterozygous and homozygous variation over there. Amarjit told me this is my favorite topic, but I will just like to share you a story why I'm interested in homocysteine. My entire family is having a homozygous and heterozygous variation in MTHFR genes. So my grandfather, who died early at the age of 57, I have a four uncle, two uncle, they are died at the age of 40 and 50, and we have cardiovascular disease in our family. My other family, my father and my other uncle, we have a heterozygous variation. So other family has a homozygous, they're very poor methylating, and their gene expression is very bad, so they get the disease early. My family, my father family, and my uncle family, we have heterozygous variation in MTHFR. So we already have a disease, but we have delayed expression of genetics. So how we can prevent the disease? Just we have to improve the methylation. We have to optimize the methylation. And how we can optimize the methylation? That's the million-dollar question. How we can optimize methylation? How we can work and how we can make the cell work that have a proper methylation. So in short, what we learn? We learn that environmental trigger, genetic expression, that's methylation is playing a role. You can prevent the disease by controlling the genetic expression. That's the methylation. Methylation. What you need, guys? We need methyl donor in our diet. That's a very simple secret.
10 minutes to go.
Because we have a deficiency of methyl donor in our diet, that's why we have a genetic expression. That's why you see a lot of chronic disease right now in the world in India. So we need methyl donor in our food. If our food is not sufficient in methyl donor, we need to take a supplements. But what kind of supplements you take that optimize your methylation? Make sure you all check your homocysteine level. Keep it less than 7. If your homocysteine level is more than 7, you should do these vitamins. You should optimize your lifestyle. Of course, we in functional medicine, we talk about lifestyle. So you can optimize, you have to optimize your vitamin D level, you have to optimize your sleep, you have to optimize your food, you have to optimize your microbiome, you have to optimize so many things. But for methylation, you have to optimize your methyl donor, and that's your methyl donor. You need active folate, you need active B12, you need active B2, you need active B6, you need zinc, you need magnesium, and finally you need TMG, that is trimethyl glycine. So it's a 3 molecule of methyl donor or methyl with the glycine that you need. So you can optimize your methylation by supplying these active forms, and make sure you understand the simple science that you need active forms, because your patient or yourself having a methylation defect, so you cannot convert these inactive vitamins to the active vitamins, and that's why you need active forms of vitamin. So these are the most common mistakes the clinician does. They prescribe the inactive forms and they expect their methylation to be a proper. We require methylation donor. So that's about the methylation. So you need to just increase your level of all the B vitamins plus magnesium plus zinc, and that way you can optimize your methylation cycle. Apart from food, food is deficient in all these B vitamins, so you need to take in supplements. Particularly if your patient have a genetic variation like me, I have to keep my B12 on upper side normal. That is my optimal range. So also in a conventional range, we are doing a mistake. We are not personalizing patient's biomarker. We are not personalizing patient's parameter. So in case somebody having an SNAP, although Dr. T will talk about genes and genetic expression, but here, when we have a genetic variation, the range of this B12, the range of folate is changed for that particular patient. For myself, I cannot keep my B12 to 400. I cannot keep my folate to less than 18 or 20. I have to keep it high because I need all the time this methyl donor so my enzyme or gene can work better. Of course, I have to work on a lifestyle, but the rest of things I have to manage. So that's about methylation, and you need to take care of your methylation to make your cell healthy and functional. Thank you very much.
Thank you so much, Dr. Musa. Indeed, everyone says they love the way you explain those complex things and make them simpler.